Okay, wonderful.
Thank
Yeah, so thanks, Sue, and to the Brain Tumor Foundation for allowing me to talk to everybody today. Um, uh, I'm going to talk about what we do, or one of the projects that is taking place in my lab, which is in Calgary. I'm a neurosurgeon in Calgary and have a lab that studies glioma, predominantly adult gliomas and glioblastoma. And I've been very fortunate to be supported by the Brain Tumor Foundation of Canada as well. And this is some of the work that's come from that support. My lab is focused on studying models of glioblastoma predominantly, and using things like brain tumor initiating cells. But what I'm going to talk about today is a project that has in part used these cells, but it has been focused on trying to understand the immune response in adult glioblastoma. And so the title today, as you can see, is IDH mutation is associated with the pro-inflammatory microglia and macrophage phenotypes in glioblastoma. And I'll describe a little bit more about that. Here we go. So I think many people here know about glioblastoma. I'll say a few words briefly about it. It's the most common and most malignant primary adult brain tumor. This is a typical MRI picture of glioblastoma. And this is really a devastating disease. It's also a heterogeneous disease, and it's something that we've learned a lot about over the last number of decades, and particularly in the last decade with the introduction of genomic technologies. But despite what we've learned about it at the genomic level, it really hasn't yet translated into any meaningful advances in therapy. But what we do understand now is that there really are genetic subtypes of glioblastoma, and that is important for understanding the biology and prognosis in patients. What we do know is that surgery followed by radiation and chemotherapy still are the best treatment measures, but the average survival, despite those aggressive interventions, is still little over a year. And the inevitability for patients with this disease after aggressive treatment is recurrence at some time, which is unpredictable. Now, I'll shift gears just a little bit. There's been significant interest and advances in immunotherapy for systemic cancers. This really was led by work in melanoma, and it's been applied now to many other cancers, lung cancers, renal cancers, pretty much every other systemic cancer. And it's been, of course, of significant interest in glioblastoma as well. And they've made dramatic advances, as probably many of us have heard, in the treatment of systemic cancers with these immunotherapy agents. Now, unfortunately, to this point in time, none of these agents have really demonstrated much impact in glioblastoma. And, in addition to these agents, there are other things, as Dr. Narendran talked about, things like vaccines and other types of immunotherapies. But, despite a lot of work, we haven't had any treatment breakthroughs in glioma. But it's led to more work trying to understand the immune response in this disease, and that's what we've been trying to do. And what we can see here is just an example. Now with the genomic and single cell type technologies that are available, we can clearly see there is some type of immune response in glioblastoma. And then in terms of immune responses, you can have um, T cell based responses, which are the predominance for systemic cancers. And so many of the therapies are directed towards T cells and reactivating those. The problem with brain tumors is there seem to be very few of these um, cells within the tumor. So then another big question is, what are the cellular components of the immune response in tumors? And what we begin to see is that it's an innate immune response with cells called microglia, which are brain derived, and macrophages, which are systemically derived but seem to infiltrate these tumors. And as mentioned, there's been a lot of work trying to look at the immune response, but overall, we found that it's very difficult to make heads or tails of that. Um, and there's been a lot of heterogeneity in trying to understand that. And then one of the other big issues that we thought about was these tumors themselves are heterogeneous, and none of the studies thus far have really looked at where the tissue comes from these tumors, whether it's from the center, the necrotic area of the tumor, whether it's from actual proliferating and highly cellular areas of the tumor, or beyond into the brain. And so what we really wanted to do is try to begin to understand what is this immune response in patients with glioblastoma. What are the cellular components? And what does this immune environment look like? There's been suggestions that the immune response is very anti-inflammatory, and then thought to allow the tumor to proliferate unchecked. But we wondered whether that was truly what was going on, because the immune response, as we know in many cancers, is actually trying to attack the cancer. And we wanted to try to understand this a little bit better. And what we've ended up focusing on, mostly just because of their abundance, cellular abundance, is this, what we call a GAM response, or glioblastoma associated microglia and macrophage response. And then trying to understand, is this a pro-inflammatory or an anti-inflammatory milieu that these cells create within tumors? And our overall question was very simple. Initially, it was, what is the immune environment like between different patients with glioblastomas? And as we got into this, we then also began to stratify this and look and ask, in glioblastomas that have a fundamental genetic difference in their underpinning, and that's one of the main branch points now in our understanding of glioblastoma, and that's something called the IDH mutation. We wanted to understand, is there a difference between IDH mutant and IDH wild type glioblastomas? The IDH mutants are by far the most common type of adult glioblastoma. They're much more aggressive, and they're the ones that we typically think of with about a 1-year survival. But we know that there's a smaller proportion of IDH mutant, the IDH wild types are the bad ones. Sorry if I said that incorrectly. And then there are the IDH mutants, which are what we see in younger people generally, and they're a much more favorable subtype of glioblastoma. So, we set out with the aims to determine the composition of the immune response in glioblastoma, to characterize the innate component, and then really to understand the activation status of these microglia and macrophages within glioblastomas. And I'll just show a few pictures of this. One of the markers, this thing called IBA1, which is a marker of microglia. And the two panels on the left really show the two ends of the spectrum of an immune response in different adult IDH wild type glioblastomas. And what we see is that on the very far left, some patients have almost a negligible immune response, or composition of cells from the immune system. And then others seem to have a very robust immune response or composition of microglia within these adult glioblastomas. And then on the very far right, what we see is an example of an IDH mutant glioblastoma. And what we saw on average is that the IDH mutants seem to have a much lower number of infiltrating immune cells, predominantly microglia and macrophages. What I don't show here is pictures of all the different cell types that we looked at, and we did look, at many of the immune cell types. And the important thing that we saw was that you rarely saw T cells in any of these glioblastomas. The immune response was almost always microglia and macrophages, and we never saw a frequency of T cells above 1%. Of this cellular a more reproducible, or not reproducible, but consistently low level of immune cells in IDH mutant glioblastomas. So the IDH wild types have significant variability. There's much less variability in the IDH mutants, but also a much lower number of immune cells. And we're not really sure what the reason for that is, other than the IDH mutants probably do grow in the brain at a much more slow rate, and perhaps induce less of an immune response than the IDH wild types, which typically grow much more quickly and probably haven't been in the brain for anywhere near to as long as the mutants. And the other important note I should make from this is that for all of these samples, we selected these directly in the operating room from the aggressive part of the tumor, or what we would call the enhancing part of the tumor on the MRI scan. So the sampling was very consistent across all of the glioblastomas. So what we then tried, and is this immune response pro-inflammatory or anti-inflammatory? And what we did to look at that was we looked basically at a number of markers of macrophage activation status. We looked at a number of cytokines, and then we looked at morphology, which is thought to correspond to activation status of microglia and macrophages, and then size of microglia and macrophages. And all of these things we put together and put into a score to look at state of pro or anti-inflammatory status of a tumor. And what we found was quite interesting. And what it basically said from one tumor to the next was that a tumor seemed to have not either a pro or anti-inflammatory status, but they seem to have either a high level of both pro and anti-inflammatory appearing cytokines and markers, or a very low level. So it was either seemingly a high level of an immune response or not. But overall, when we put this all together, what we found was that the IDH mutants, despite having a lower number of innate immune cells, seem to have a much more pro-inflammatory nature of this immune response compared to the IDH wild types, which seem to be less pro or more anti-inflammatory compared to the mutants. And so we thought this was quite interesting. The number down here just reflects our patients and the survival difference between the IDH mutants with average survival just over a year, 368 days, versus the IDH wild types, which have a dramatically better survival there. And that's been known now for some time. And then finally, what we then did was try to confirm this, and we looked at publicly available genomic databases which have done single cell sequencing, and then tried to understand what are the cellular populations in IDH wild type compared to mutant glioblastomas, and does this confirm what we saw at the immunohistochemical and cytokine level? And really what this ended up showing us was that in the IDH mutants, their immune response was predominated, or the predominant cell type were microglia, as we knew. And so they were also pro-inflammatory. So the IDH mutants are really characterized by a pro-inflammatory microglial or innate brain-derived immune response. And again, the IDH wild types are much more heterogeneous, with both macrophages and microglia and variation along the pro to anti-inflammatory spectrum. So in summary, the immune microenvironment really is very different from patient to patient, particularly in IDH wild type glioblastoma, the most common type. The immune microenvironment does differ depending on IDH mutational status, with the IDH mutants having a lower number of immune cells, but with a much more pro-inflammatory nature to them. And then we feel that beginning to understand the immune microenvironment and the heterogeneity from patient to patient may begin to allow us to understand therapeutic vulnerabilities, and and hopefully this will improve our understanding and better the way for immune type therapies, which have proved so beneficial in systemic cancers. And one way that we're now pursuing is thinking about how to alter the microenvironment to better treat these glioblastomas in the laboratory setting. So with that, I'd just like to thank the members of my lab and a number of other labs, one Wei Yong's lab, Greg Cairncross's lab, and Jennifer Chan's lab, who've all helped with this work. And then special thanks to Candace Poon, who's a very bright young neurosurgery resident who recently completed her PhD in my lab and is responsible for this work. And then, of course, I'd like to thank the Brain Tumour Foundation again for providing support for this research in my lab.
Thank you very much, Dr. Kelly, for taking the time to present. Somehow I seem to not be able to see my chat options. Let me see if I can change that. I'm not sure why they've switched. Can you see the chat, Dr. Kelly?
Let me just see. Yeah, I can see it now.
Okay, so in the meantime, if anyone asks any questions, I'll leave that to you while I'm trying to figure out why I can no longer see it. So for anyone that has questions, if you can just type them into that chat box. So Dr. Kelly, do most patients find out whether their tumor is wild type or not, and how do they find that out?
In Calgary, certainly we communicate that, or I certainly try to communicate that to all of our patients. And I think they would do that at the neuro-oncology clinic in Calgary. I'm not sure if, I'm not sure how that varies across the country, but I would think that most centers now test for that. And so I think that information should be readily available.
Thank you. Any other questions at all for Dr. Kelly? Hi, Dr. Kelly. Great presentation. Is there a reason why T cells are such a low level in the brain?
Well, I don't think anybody knows that for certain. The T cells are part of the systemic circulating immune response, and it's known that T cells in number or the number, in a normal individual are extremely low in the brain. And the brain is known to have this innate immune system, which is made up of microglia predominantly. So I think it's just a difference between the brain and the systemic immune response.
Thank you. Any other questions for Dr. Kelly? Can we irrigate and inject tumor? Oh, hold on.
Irrigate and inject tumor bed during surgery. I'd say the answer to that is yes. I mean, we irrigate the tumor bed with sterile saline solution during surgery. I mean, you could irrigate it with whatever you wanted, and then there's a type of treatment called convection enhanced delivery, which has been tried time and time again with glioblastoma and various therapeutic agents. I think at this point in time, it just hasn't been demonstrated to be effective to inject various chemotherapeutics or various other types of drugs. There certainly are trials ongoing with convection enhanced delivery and delivery of targeted toxins, things like IL-4, IL-13, conjugated Pseudomonas exotoxin type things. So people have thought of and tried this approach, but it just hasn't demonstrated to be effective yet.
Thank you. I enjoyed your presentation very much. I agree. IDH mutation status is available pretty much all of the time. How can you modify the responses of microglia systemically?
Well, that's what we're trying to understand now. And, there have been a couple of publications, mainly done out of Wei Yong's lab that we've been part of as well, where, a few years ago, they showed that antifungal agent, amphotericin B, could do this. There's another recent paper from his group that we've been part of where they use niacin that also seems to alter the innate immune response by systemic administration. And then we're also looking at a number of other things to do this systemically, but both topically at the tumor bed site after resection in animal models, of course. So through various agents like that can be administered systemically, they do seem to have an intracranial response on the microglia. And then your second question, does IDH mutation status change at recurrence? That's an interesting question. I think the easy answer to that is no, it doesn't. But what we observed in the lab when we have grown IDH mutant tumors is that, interestingly, sometimes we would grow them and they would lack the IDH mutation. And what we found then through karyotyping the cells was that what the tumors seemed to do as they evolved over time was they lost the chromosome that had the allelic loss of IDH mutation, making it look like a wild type tumor, but really it had just kicked out that portion of the chromosome with the mutation. I haven't ever seen that happen though in the clinical setting. So I'd say the simple answer is no, it doesn't change.
Is there any further questions? Okay, excellent. Well, thank you very much, Dr. Kelly, for taking the time and for joining us in this modified format. I want to thank everyone for participating today and still being able to join us in this modified version. Again, the presentations have been recorded, so we will be posting those eventually. As you can appreciate, there's a lot of things that we're modifying at this time, so our team is frantically working, so we will do the best that we can to get those up for everyone to view. Just a couple of reminders again that this is Brain Tumor Awareness Month, so please spread the awareness, spread the word. And for anyone who hasn't registered and is interested in registering, we would appreciate your support in joining the our walk and the 27 Canadians Challenge by asking 27 of your friends to give $10 or 10 of your friends to give $27 or whatever that looks like. We would love your support. And obviously in the times that have presently changed, we really want the brain tumor community to know that we're all out there, we're all rallying for everyone, and we have seen that today, and seeing that research is still happening, and different fundraisers are perhaps being modified or put on hold to support our programs and services and the research that we offer. And I thank you all. It truly is a true testament to say that we are all in this together. And all of those who might hear those words today, that they've been diagnosed with a brain tumor, we need to show the support that we can, and all of the awareness that we can spread about this devastating disease. So thank you so much. This concludes our first Brain Tumor Research Symposium virtually. So thank you again, and I hope that you all have a wonderful remainder of your weekend.